I would like to first stress that you are welcome to ask me any questions about antibodies and Western blot. I've been working with the antibodies and Western blot technique for over 25 years. I will be happy to answer your questions. You can write them in a chat box, and I will take a look at them during the break. So first, a little bit of the introduction of where are we from. Agrisera is located quite far up north in Europe. So at the moment, our days are a little bit dark, but in the summer, we experience just constant light, which we enjoy very much. And we have two facilities, which are located, one outside Umeå, where we are producing polyclonal antibodies, and the other one, which you see on a picture, located in Umeå, where we are conducting antibody purification, quality control, and shipment. And on all the pictures, you will see our staff. They are not taken from Shutterstock. The company has been around for quite a while, as you see, and we have become a part of Olink Proteomics in 2020, and we are now producing polyclonal antibodies for their proteomic platforms. However, our polyclonal antibodies for plant and algal research are our original brand, which we developed over the last 25 years. We can support you also with extensive collection of epitope tag antibodies, including Turbo ID, as well as matching secondary antibodies of high titer and detection reagents. But now let's concentrate on what we came here for today, the Western blot. I wonder if you're aware how old that technique really is. Think a number, a year in your head, and I will show you where did it come up that researchers found out how to visualize specific protein in a complex protein mix. It was actually the year 1979, so the technique has been around for quite a while. And the name Western blot comes from the rows of winds, and is a little bit of coincidence. You're probably familiar with Southern blot for DNA, Northern blot for RNA, then we have Eastern blot for post-translational modifications, and a Western blot for proteins. And Western blot comes from the West Bank, where the lab which first published the article on Western blot was located. Just a short reminder about the principle of a technique. We are first separating electrophoretically proteins in a gel based on molecular weight, and afterwards we're transferring them to a membrane. Can be one of a transfer type. You see here is a sandwich. We apply electrical current, and the proteins are migrating from a gel to the membrane. And this is so-called transfer stack, which you are using if you are using wet blocking. Always remember to use highest quality reagents, of course. The goal of protein transfer is to obtain exact copy of what we had on the gel being transferred to the protein, to the membrane, especially in the region of your target protein. This is a principle, and how it is conducted is very important. I will show you different aspects of Western blot, what can go wrong and what to think about before you even go to the lab. But at first, a little bit of basics. Antibody is a Y-structured molecule, and the most important part you see marked as this in yellow antigen binding domain. This is a variable part of the antibody. This is where the binding to your target protein is occurring. And we have this region where the antigen binding domain is located is called FAB region, but we have also FC region, a handle fragment that crystallized which is called like that because researchers left the solution in the lab, came in the morning, and it was crystals. And that part is crystallizing the name FC. So this is how the antibody derived from goat, mouse, rabbit is looking. You may be also familiar with antibodies coming from camelids like llamas. They are a little bit smaller and can more easily penetrate, for example, in a tissue when we are conducting immunohistochemistry experiments. We will share the presentation with you, and it contains a lot of different links to further blog posts. So please do not worry, you will be receiving all this material. What is an epitope? That's a very important part to remind you. We have so-called linear epitopes. As you see here in the antigen binding domain, the antibody is binding to linear sequence of amino acids. The smallest epitope is 3 amino acids, up to 15 and more. So in Western blot technique, in techniques where proteins are denatured, then antibodies are binding to linear, so-called linear epitopes. But we are also having a 3-dimensional epitopes. And these are created by the protein fold when distant amino acids are brought together. And these continuous epitopes are mainly recognized in techniques where antibodies are binding to native protein, which is folded. This is very important for understanding how Western blot is working, how antibodies are working. And also, I would like to remind you about the clonality. Antibodies can be poly, means many, or monoclonal. Polyclonal antibodies, it means that there will be a pool of different antibody molecules which are binding to different epitopes, consisting of at least 3 up to 15 and more amino acids if it is a linear epitope. And they can be derived from, antibodies can be derived from different species, here in the order of how much of the antibody we can obtain. Mono means one. Monoclonal antibody will recognize only one epitope. And it is a little bit of a myth that monoclonal antibodies are always better. I would not agree with it. To my experience, polyclonal antibodies are more versatile. They recognize more epitopes and can be used in a wider range of techniques. Of course, there may be also increased background signal, but I will show you how to deal with it. If a monoclonal antibody is losing activity, you will not get a signal from this antibody any longer. If it is a polyclonal antibody, you may see deterioration of a signal. And also, please keep in mind that monoclonal antibodies are targeting only one epitope. So if it is an epitope which is buried inside the protein fold, such antibody will not be suitable for techniques where, like CHIP or other techniques where we're dealing with detection of a native protein. So remember, antibodies are binding to so-called epitopes, which can be linear or conformational. And antibody choice must match species and application. And this is the most common mistake which I see is done, that we are assuming that a given antibody is going to work without actually checking it on a sequence level. This is possible to predict, and each year I'm finding a couple of publications where Agrisera antibodies have been used on the wrong species, but still a figure with a nice band is presented. So please keep in mind, it is very easy to obtain false positive results with the antibodies. Therefore, before you go to the lab, you should actually check if the antigen which was used to elicit given antibody is found in a protein you're aiming to visualize. We have developed a panel of so-called global antibodies. So this means that short peptide, the shortest peptide we used is 7 amino acids and is found in the target protein coming from all these different species. So global antibodies will have a wide taxonomic range, but this is not the case for always. So I will show you how to actually check if the antibody you intend to use is correct. Is it the right antibody? Antibodies are developed to peptides to make them more monospecific because they will target, as you see in this example, a peptide at N-terminal, C-terminal, or inside a protein. Of course, we need to target a peptide after signal sequence is cut off. So these unique peptides are unique to that specific protein and should not be found in any other proteins coming from that species. In our design process, we're blasting them towards the proteome of the species to confirm that the peptide is absolutely unique. So if the antibody have been developed to the peptide, if full-length sequences are aligned from the protein you aim to detect and the protein the antibody is made to, it will be irrelevant. If product info sheet does not include this sequence information, you can always write to the supplier and ask them and provide them the sequence which you aim to detect. So please keep in mind that overall sequence homology is irrelevant if we are using short peptides for antibody development. And here, what do I mean that 70% of linear sequence should have the identity? It happens that if sometimes 10 out of 15 amino acids are conserved, that may be enough for the antibody to work on another species. And this question I am receiving quite often. Can knowing antigen sequence help me to identify cross-reacting bands? Unfortunately not, because choosing antigen sequence is unique for a given protein. As I mentioned, we always check it and blast it. So you can identify cross-reacting bands only by mass spec or sequencing. There is absolutely no other way to do it, unless you have specific mutants. This question I have also received, and therefore I'm sharing this information with you, as we are developing antibodies to plant proteins. And if you aim to develop your custom antibody with some company in the future, please keep in mind that the serum from antibodies which are not from animals which have not been immunized need to be checked before immunization. In this example, I will show you a Western blot, which is done on Arabidopsis and barley. And we have tested as using as an antibody, primary antibody, is pre-immune serum from these animals. And as you see here, before immunization, the antibody, the pre-immune serum is already recognizing proteins in plant extracts. Especially famous may be Rubisco. So with this screening, we would not use the animal for antibody production if we see that there are already bands in the target protein range. This is why if you are getting the antibody from somewhere else, and this has not been done, that you may see different cross-reactions coming up. So, for example, the animal number 7, we would choose for production of antibodies to proteins in a high molecular weight range. So this is an important step before even an antibody is produced. And now just a short reminder how the binding is occurring. So you separated the protein in a gel and transferred it to a membrane. The membrane is blocked. And then we have a primary antibody, which is binding to your specific target protein. And we are applying a secondary antibody, which is labeled with enzyme or fluorophore, and this gives us possibility to visualize our unique protein. This is one of the options, and as you see here, there are two antibody molecules of a secondary antibody binding, and the secondary antibody gives an enhancement of the binding. Another option can be used when primary antibody is directly labeled, but this option I would rather recommend you for proteins of high and medium abundance. Because for proteins of low abundance, like transcription factors, we may require this enhancement of the signal. So this Western blot is composed of quite many steps, and hopefully once we get through them, the result is as nice as here. I will guide you through this process now and provide with information which I hope you were not aware of. The theory is good. How about the practice? What you see here are Western blot examples from labs which were receiving the same primary antibody to a transcription factor P4, and as you see, the outcome was very different. One of the laboratories hit the optimal conditions for protein visualization, which may also include extraction. For some proteins, extraction must be done, for example, in the yellow light, almost in the darkness, because otherwise they will degrade. So this has to be taken into account before you even enter the lab and start doing the Western blots. So ask yourself, how much do you know about the protein you are aiming to detect? And what do I mean by that? Protein checklist contains biochemical properties, including isoelectric point, which will influence protein solubility, of course. And expression level. Is my protein of high or low expression? Where is it localized? In which organ, which tissue? Does it have specific cellular localization? When you may ask, why, how can I know the level of expression of my protein? There are actually the databases where you can obtain this information. My favorite is PAX, Protein Abundance Database. I'm using it daily. It contains data for not only Arabidopsis, but also other plant species. And as a rule of thumb, if a target protein is listed as abundant less in 10 ppm, we consider it as difficult to make a useful antibody and facilitate detection. So please keep this in mind before you even enter the lab. I would strongly recommend to check that, to check, am I dealing with abundant protein or not? The other database is from University of Toronto, is providing the mRNA data, which can also be useful, but of course, mRNA is not always translated fully to functional protein. And then your sample need to be, of course, of high quality. If you would like to use these images, they can be downloaded from our website. There are different extraction methods, and some are more or less time-consuming. For multiple samples, I would like to recommend you to consider the bead beater, where you can process multiple samples at the same time. When we're doing mortar and pestle, which is a good extraction method, but maybe 20 samples cannot be extracted in the same day. Sonication is prone to heat generation. So depending upon how many samples you have, you need to consider the most suitable extraction method for your protein. We summarized it in our newest educational poster, which you can download from our website or request a hard copy for the wall in your laboratory, and we will be happy to send it. And for proteins of low expression, please look at this example. If proteins are of low expression, you may not be able to detect them in a total cell extract. Quite many antibodies have been discarded at that stage because there was no band. But if protein is of low abundance, fractionation is really necessary. As you see here, by fractionating, we concentrated target protein. You may ask me what to do if a target protein is cytoplasmic. Yeah, then in such case, there are precipitation methods which can be used to concentrate your target protein. This is very important, and I know that fractionation is a laborious process, but it may be worth it. There are also different kits which can aid you to do that. And extraction method can give you different outcomes depending upon buffer composition. So please see here, that was our test lab, which was testing BAC1 antibody, and with HIPI's buffer, they obtain a target protein band, but not with the other buffer. The buffer composition may be important and crucial not for all proteins, but there are some which will extract better depending what are actually extraction buffer components. And here in this example, it is so-called garden blot. So we have barley, Arabidopsis, and rice. And we knew that the peptide which was used to elicit this antibody is found in all these species. And why do we not have a band? Well, proteins may partially refold following Western blot. So in this case, we applied 6 molar urea and strong denaturant to unfold the protein, and the band was visualized. There can be also mobility difference depending upon from which species a target protein is coming. In some cases, you may consider extracting a protein from very specific organ, for example, 2 millimeter root tip. If it is a pin protein, you need to include protease inhibitors unless you're doing your extractions extremely fast. In some cases, even proteasome inhibitors. We had a case of a lab which was really trying to visualize the target protein, put a lot of effort in their experiments. ELISA data were looking good, but there was no signal. And it appeared that the protein was a target of proteasomal degradation, the protein they were aiming to detect. So adding a proteasome inhibitor, MG132, helped to obtain a target protein band. So you need to consider first what extraction protocol should be used. And each protein may be a little bit different. So please use the checklist which I have showed you, because some proteins may require very specific extraction conditions. And how do we work? How should we work when we're working with the Western blot? Here you will see a shortening GLP. I bet some of you know what it means, good laboratory practice. And what do I mean with that? We are working clean. It is very important in Western blot procedure that all equipment and all chemicals are of highest quality. We're also working in the cold, so we are using cold buffers. And if we're not running the electrophoresis and transfer in a cold room, we do it with cold buffers. We are precise. So ask yourself, when did you calibrate your pipettes last time? We are using fresh reagent stock. We do not reuse the buffers, and we follow protocol. So when it's written 10 milliliters, it's 10 milliliters. And hopefully then the results are going to reflect our good laboratory practice. I received the question, why should we use precast gels? Precast gels can be used in the last stages, like for example, if you want to refine your results for publication. As you see here, there can be difference between using tris glycine and tris acetate gels for high molecular weight proteins. Precast gels may offer you different molecular weight range, and quite good shelf life. And also, if you're doing any quantifications, they may be quite helpful. This is one of examples when the gel was not, it was a different pH in stacking and resolving gels, and therefore such an outcome. So precast gels will be produced in controlled conditions, help to avoid these kind of issues. Another question I received, what should we load per well? And I wonder what you are loading. Is it protein, chlorophyll, or volume? We strongly recommend to load on protein and on microgram of chlorophyll for photosynthetic tissues. We do not recommend to load on volume. Why? Because especially if you are working with low abundancy protein, if you load on volume, you do not really know how much of your protein, how much of a protein you have in a well. And it can be of importance if you need to load quite a lot. Because for some proteins, to detect them, we need to load more than 50 microgram per well. There are reagents which will make it possible for you to estimate protein concentration in presence of components like denaturing reagents, SDS, because I often hear that, well, it's not possible to measure protein concentration in a loading buffer. And actually that's not correct, because it's possible. It adds an extra step, but it is worth an effort. And should it be nanograms or micrograms per well? And this also depends. If you're dealing with recombinant protein, then you're loading nanograms. When it is an extract, then you're loading micrograms. And here we have a titration when we have same protein load, but we are diluting the background by using primary antibody more diluted. And here we can titrate for the publication to see at which combination of protein load per well and primary antibody dilution the background will be low. These are like refinements of Western blots when you have them working. That was also one of the questions which I received. Should we use 1D or 2D gels? Just a reminder how they are looking. So in 1D gels, we are separating proteins based on molecular weight, and each such band, which we see, may contain hundreds of proteins. So in a second dimension, each such band is further separated to spots. I am mostly, 99% of my troubleshooting are one-dimensional gels, although I have seen also results of Western blots done on 2D gels, but they differ between each other. That's just a list for your reference, but it of course depends what biological questions you're aiming to answer. Now we're coming to sample denaturation. It can be done in different temperatures. I bet you are using one of those. We are recommending to use 70 degrees for 5 minutes because some proteins may aggregate when they are heated excessively. For example, H+ ATPase, membrane protein, which will aggregate and not enter the gel. So that can be a reason for lack of a band, because the protein aggregated and is stuck in the top of a well. These conditions are also used in certain specific cases of low temperature denaturation. In general, 70 degrees for 5 minutes is enough. And how do we do our transfers? This is a picture from our lab. It's a nice bucket we are using for protein transfer. And how important are conditions of a transfer? These were the questions I have received. And there is actually one important point you have to consider. It's molecular weight of your target protein. So before you go and do the transfer, you need to do this short check, because proteins below 20 kilodaltons will migrate faster from the gel, so they will require shorter time. I've troubleshooted many blots where there were difficulties to detect protein, which was around 9 kilodalton, and the reason for it was that it was blasted through the membrane. So the transfer was done too long, and the protein was left in the buffer and didn't stay on the membrane. Therefore, there was no band. So recommended also are membranes with 0.2 micrometer pore size. And for high molecular weight proteins, we use membranes with 0.44 micrometer. They also may require more SDS, and it is generalized to say that high molecular weight proteins transfer better in wet tank. Wet transfer is less prone to failure. However, it may work as well good independent of if it is wet or semi-dry transfer, because it will more depend upon the protein and the membrane type you're using and how carefully you're conducting whole procedure. But there can be sometimes differences that with certain protocols, the results will be better. Which membrane to use? Yeah, I bet that you're using one of those, PVDF or nitrocellulose. What I can see is that PVDF is more common. It has another mechanism of binding compared to nitrocellulose, can be air dry and rewetted, and has a better protein retention, is more robust. However, nitrocellulose can be also air dried and rewetted, but it's more fragile, so it will be more easy to break it. And both membranes are available in these two different pore sizes. So I have seen over the years that some proteins, protein antibody pairs, may give better results with nitrocellulose than PVDF. And this is what we are writing also on our product info sheets to save you the work. And membrane activation, I received a question, methanol or ethanol? First choice is methanol because it gives more efficient activating of the membrane and facilitating of protein binding. If health is of concern, ethanol can be used, but you need to keep in mind that it will be less effective. And now we are moving into transfer type. So is it any difference between efficiency between semi-dry and wet? Of course, they differ in amount of buffer, electrode contact, and speed. But all also depends how you conduct it. So it can be an excellent protocol, excellent setup, but for some proteins, wet transfer may actually work better than the semi-dry. So it is not written in stone that one is better than the other. It has to be tested. How to efficiently remove air bubbles when we are working with the protein transfer? Please, we are not doing like that with the roller. We have abandoned this method. Instead, we are assembling the sandwich directly in the cassette, as we realize that this improves sharpness of the bands, and the sandwich has to be assembled tightly. So if your pads are old, you can add an extra pad, and do not move the sandwich too much. That's the key. We have made a short video, which you can later watch if you would like to. And of course, air bubbles are important to be removed from the areas of your target protein, because they will be influencing protein transfer. If we have an air bubble at the top, we will gently try to squeeze it out, but it will not be that crucial. The most important is the area where your target protein is. That area should be free from air bubbles. We are making a quality control of a transfer can be done using either Ponceau or Comasi. 250. And if we see that our blots look like that one here, then we're not going to continue with it. Something went wrong, and this is going to be a waste of time to continue with such membrane. The even more important quality control is to stain your gel with Kumasi. It is very quickly done, and I would like to recommend you to do it every time you're doing the blotting. And why? Because you can see if all proteins left the gel. Here in this example, we have been doing a sandwich where we had 2 gels and 2 membranes, right? And of course the pads. And as you see here, the proteins were transferred from the gel 1 quite effectively, while they were left behind in the gel 2. So we decided that for quantitative work, we need to have an even protein transfer. Therefore, we are using only 1 gel per cassette. So quick and easy, if you see any protein left in your gel, it means that your transfer is not good enough. It has to be extended. When we come to primary antibody format, the antibodies which you're using can be in those 3 formats: as serum, as purified to total immunoglobulin using protein A and G, and the part of antibody which is not changing, which is called FC. This is where the binding occurs. And they can also be purified on specific antigens. And when you're reading product info sheets, please pay attention to antibody format, because it may look like a lot of antibody which you're buying, but if it is in a total immunoglobulin format, it will still be only 0.5 to 5% of what you have in a tube will be a specific antibody. I'm often getting questions how much of specific antibody is in serum. It cannot be determined until antibody is purified on the antigen. So the best are antibodies which are purified on the antigens. Well, the best, you will have the most antibody per tube. And the purification is done in that way. Oh, sorry, I was too quick. We're using chromatography equipment. We have columns where we are coupling the antigen, so it can be a protein or a peptide. We are first coupling it on the beads, and then we're passing the serum. Specific antibodies will bind, the rest will go through, and then elution is done usually with low pH, and the specific antibody fraction is obtained. So the question about the antibody, which I also often receive, is, can it be reused? And I'm curious if you are reusing your antibodies, because this is quite a common practice. They are expensive. I understand that. So how long can the same aliquot of antibodies in blocking buffer kept in -20 be used? And that can depend upon the antibody. So some antibodies can be used once, while the others can be used quite many times. But you should not be too disappointed if you reuse your antibody and it doesn't work, because it depends upon the antibody. It means that all specific antibody bound to target in the first incubation, there's nothing left. Therefore, in the second blot, you have to accept that that specific antibody cannot be reused. So please keep this in mind for your future experiments. And definitely, if you're working with antibody which is not characterized, you should never reuse it, because you will not know if the outcome of the second Western blot, which is negative, is due to antibody not being able to be reused, or if there are other factors. So here, there should be some carefulness. And primary antibody does not work any longer. What has happened? We had seen antibodies which are stable for the last 40 years, and we have also seen antibodies which lost activity after 1 year. And what does it depend upon? This is the antibody binding domain, which is different depending to which epitope the antibody is binding. And this will influence the stability of the antibody. So one cannot really predict how long the antibody may last, but can be different reasons for it. And the storage temperature, if you had freezer issues, that's the most common one. Purity grade. Actually, antibodies in serum are protected and surrounded by different proteins, while when they are antigen affinity purified, they may be actually less stable. So never purify all serum to obtain your antigen purified antibody. Lack of stabilizers such as glycerol or BSA, although one should not add glycerol to serum. This is absolutely no need for that. If you use the same antibody multiple times, you can also see deterioration of a signal. So that's a sign that this should not be done. And also not following recommendations on product information sheets. We have always stored antibodies minus 80, but the antibody you purchased maybe is deteriorating minus 80. So always refer to product information sheet, because each antibody may require a little bit specific storage conditions. I have been storing chicken antibodies in room temperature for 1 year, and they were performing pretty well. So depending upon the antibody, not always minus 80 freezer is the best solution. How to remove unwanted background. I think many of you experienced this issue in your Western blots. Here it was an interesting case because the antibody was weakly recognizing target protein, and the customer was wondering if it's possible to clean it up somehow. And actually it is, because if you have a mutant of your target protein, you can prepare a strip, or so load the gel with 10 samples from your mutant and transfer them to the membrane. So such a membrane will lack your target protein. And then you incubate this membrane with your dirty antibody. Afterwards, you collect the antibody solution and use it on your membrane of interest. I hope it is clear. What happens is that all the cross-reacting antibodies will bind, but as your target protein is not present on that membrane, these antibodies will remain in the solution. And this is a very common method to purify, for example, away antibodies to Robisco. They are very often a problem, especially for antibodies for people working with plant samples. As you saw on my slide on the beginning, antibodies to Robisco may already be present before immunization is starting. So that's not strange that later on they will come in quite big amounts. You may require to do 2 rounds of such cleaning to see a visible effect if you're working with Robisco. I have also described it in this blog post for your future reference if you're curious. And now we are diving deeper into the blocking. Do not use tweezers as we did. The tweezers should be having flat ends, and the membrane should always be taken care and not being left without a buffer. Your Western blot membrane should always be covered by a solution. This is a very important point. And is blocking temperature sensitive? That was the question. Should we block in room temperature or in cold room overnight? I will answer this question shortly, but at first, I would like to tell you what happened here. We actually didn't have a primary antibody to incubate with this membrane. So after blocking, we dried it. and stored it until next week when primary antibody was available and we could continue with Western blot procedure. Normally, we are preparing our membranes in advance with different cell extracts, and we are storing them in room temperature in sealed plastic bags for up to 6 months. How did you dry your blot? Okay, I mean, you are putting it on the Whatman sheet, protein side up. This is important. And you leave it in room temperature for a few hours. So this is how your membrane, you need to be sure that the membrane is dry, and then you can store it for up to 6 months. So if you have precious samples, they may be safer on the membrane because they will not be prone to degradations. Some proteins can degrade even when they are stored in -20 degrees. So this is one possibility to store precious samples already on the membrane. And here I'm listing the blockers. Most of blots I'm troubleshooting is 5 to 10% non-fat milk is used for blocking. We have well-working commercial blockers, but they are proprietary. If you need non-protein blocker, PVP40, used for also a reagent used in fractionation can be used. We have BSA and we have gelatin. For some protein antibody pairs, maybe BSA be more efficient, but what I have seen in these hundreds of blots was actually non-fat milk, which was giving the best results. And now I would like to show you an example. The lab returned the first blot here and says antibodies unspecific. Okay, I'm checking. The blocking was done too short and the blocker was not strong enough. So please see what happens when the blocking time and concentration of a blocker was increased. And this blocking has been done in room temperature. In this example, we have a blocking done overnight. So if we are to block overnight or not depends upon the protein antibody pair. For some pairs, blockage overnight may be a better option. It has to be experimentally tested. And now membrane blocking background continues. How to reduce background signals while reprobing, stripping and reprobing. What you're doing when you are stripping and reprobing, you may remove also the target from the membrane. So please keep it in mind. And if you remove the target, then of course you may increase the background because the antibody will have less of a target to bind. Generally, I'm not fond of stripping and reprobing, and we're not doing it in our lab, trying to find other solutions. And here, comparing antibodies between each other, how does it work? Actually, that's not, because one cannot compare fully characterized primary antibody to actin with the antibody to a protein of low expression, because the signal from actin will come much faster than from a transcription factor. I mean, the abundance of these proteins is different. So please do not compare antibodies between each other because they also recognize different epitopes. So it is not a fair comparison. Loading control is often used, and one should be careful with that concept because we have, of course, different proteins which we can use as loading controls, but you need to keep in mind that in certain experimental conditions, even tubulin and actin level can change. So they are not constant. And actually, to choose a proper loading control, one should make mRNA profiling and check which protein in my experimental setup is remaining constant. We are describing this concept in more details on our website. So certain consideration here is very important. Antibody incubation buffer. This was an interesting result which I have received and I would like to show you. Most of the labs is using TBST, us included, but for this protein antibody pair, PBST was giving a better result. The buffer with PBS buffer should also not be used when we're working with phosphor-specific antibodies. One should keep this in mind. And now antibody incubation. How can we save time? Actually, we can save time considerably. Please look at this example. On precious samples, the lab decided to incubate 3 primary antibodies at the same time on one membrane. It is actually possible, but you need to use well-characterized antibodies, which are directed towards target proteins, which are at least 20 kilodaltons apart in their molecular weight. And here, another method, when we can incubate everything at once, we are incubating primary and secondary antibody at the same time. And we have done this in that series of timeline here. So you can see that after 30 minutes, signal intensity was comparable to the standard method of doing steps subsequently. This can be used for proteins of high expression and well-characterized antibodies. So you can shorten your Western blot protocol considerably using this method. And a third method, when we have directly conjugated primary antibodies, like in this example, Rubisco antibody, which was directly conjugated to horseradish peroxidase, and primary antibodies can be conjugated also to fluorescent dyes and be used in, for example, immunohistochemistry.
Sorry to interrupt, Joanna.
Yeah.
We have a question in the chat. I don't know if you'd like to answer that now.
Yeah, but I answered that question already. How did you dry your...
It's in the Q&A section, and it reads, can you explain again why we can't compare antibodies to each other?
Oh, yeah, of course, I can. Well, if you take actin and transcription factor and try to compare them on the same blot, actin is much more abundant and transcription factor is not. And the actin signal will be visualized oftentimes within 1 minute, while to visualize the transcription factor signal, it might take several minutes up to half an hour, depending what reagents and what setups you're using. So in my eyes, one cannot compare antibody, first of all, specificity, because they bind to different epitopes. And then also their strength depends upon also how much target is for the antibody to bind. So therefore, I would not compare antibodies directly with each other. And also, oftentimes I hear that I purchased antibodies from the company and the antibody number 1 worked, so the antibody number 2 should work too. And that's irrelevant because they bind to different targets. So each antibody has to be optimized in its own way. And the most common mistake is to use the same protocol for all antibodies in the lab. And unfortunately, it may not work like that, especially if you are working with proteins which are a little bit more difficult. So I hope that I answered your question. Okay, so I continue with the secondary antibodies, and they can be coming from different hosts. They also look like the primary, of course. They have FAB region and FC region. And you may wonder, why is it important to even consider what secondary antibody is used? Sometimes I troubleshooted blots when incorrect secondary antibody was used. So just a reminder, we have primary antibody from the rabbit, secondary needs to be anti-rabbit. But there can be a difference in how secondary antibodies are performing. Some may be stronger, and also you should always use secondary antibodies which can be used in high dilution, because they will last for your thesis for example, but also it will minimize your background in the Western blot. And please do not exceed incubation time with secondary antibody for longer than 1 hour. It's absolutely not necessary to incubate longer, and it can contribute to background ghost bands. So 1 hour is enough. And when purchasing antibodies, we are always concentrated on how many milligrams or micrograms are in the tube. And that's actually wrong, because you should look at dilution instead of amount, because the dilution will tell you for how many experiments the antibody can be used. So that's just a reminder for the future. And now a little bit of question for you. For a blot on plant samples, which secondary antibody would you choose? The first one, goat anti-rabbit IgG, horseradish peroxidase conjugated, or the second antibody, also to rabbit IgG, but adsorbed against IgG from all these different species. So think for yourself, which one, first or the second? And the first one would be enough, because the second antibody would be more suitable to work with samples which are coming from these species. So adsorbed secondary antibodies may be of more importance when you're also dealing with immunohistochemistry. For Western blot setup, an unadsorbed secondary antibody will be enough. And now we are moving into the exciting part of detection. My favorite detection method is chemiluminescence, which requires CCD camera, and we have secondary antibodies conjugated with horseradish peroxidase. It's a very sensitive method, sometimes too sensitive. As you see here, photosynthetic protein of photosystem 2 was visualized with 2 different ECL reagents. One which is extremely sensitive, extreme low femtogram detection range, and of course the reaction gives a lot of background bands, while using the other reagent, which is suitable for proteins of medium and high abundance, they will give you a cleaner picture in a Western blot. So oftentimes background may come from using of too sensitive ECL reagents. Disadvantage with ECL is that signal needs to be recorded immediately. The membrane cannot be stored and recorded for later, and is very appropriate for quantification and analysis of proteins present in low levels. But there are some issues. You may be seeing such blots. Here's a list of possible causes of such outcome. So too concentrated or too diluted secondary antibody, sometimes interference with plastic. If you're wrapping your membranes, the plastic may actually giving a trouble. Insufficient blocking, insufficient washes, poor membrane handling, and contaminated buffer. Membrane dried during procedure can also result in such outcome. And if you cannot detect, or if you see a lot of unspecific bands, but not your target band, can happen that primary antibodies recognizing epitopes on other proteins. That's possibility depending how the antibody was made and how much of your target protein is present in analyzed sample. Target protein can also be aggregated or degraded. SDS may interfere with proteins and no target protein on the membrane, and therefore all other bands will come up. Please remember that Western blots is about signal to noise ratio. So if you have a target well exposed for the antibody, the signal from the target will be visualized very quickly before any background bands will show up. Ghost bands, maybe you have experienced that. It means that you use too much of a good thing. So too concentrated primary and secondary antibody. So the reaction goes so fast that it's exhausted very quickly. Another issue with chemiluminescence, or issue, another possibility is I receive the question, if something goes wrong, can I just redo the incubation again on the same membrane. And yes, it can be done, primary and secondary antibodies without stripping within the same day. You can save your experiment. Fluorescent detection is not used to the same degree as ECL, as far as I can see. However, more and more labs is going towards fluorescent detection. It requires, of course, an imager, and advantage, the signal is present by months, and it allows also multiplexing. As you see in this example, we have two different proteins visualized on the same blot using different secondary antibodies. Disadvantage is that the dedicated equipment is necessary, and lower sensitivity may be an issue. I have also seen incompatibility of secondary antibodies provided by suppliers. But that can be solved by changing secondary antibody. You also need to make sure that you use a proper setup, proper secondary antibody to the setup which the machine is having. And the third detection method, which is quick and inexpensive, it's a colorimetric detection method. No camera is necessary. We can record the result and store it in a lab book. Although that method is not suitable for proteins of low expression. It has too low sensitivity for that. So hopefully with these three detection methods, you're choosing the proper detection method for your protein of interest, and you're obtaining good results in the future. One thing I would like to bring up here is molecular weight markers, because I've seen that misinterpretation of results due to lack of the membrane was turned upside down, and therefore I recommend to use markers which are already having IgG binding sites and which will be visualized directly when the reaction is developed with a secondary antibody. This is very convenient. We are using magic marks, but there are different brands. We are mixing markers for gel electrophoresis with those for reaction development, and then oftentimes much less is needed per well than recommended in the product info sheets. So this is a very convenient and secure method for visualization of molecular weight. Epitope tag antibody no signal. That was also a question I received. GFP antibody, I used it on another protein expressed in our lab, and it does not work. Why is that? Well, the reasons can be different. So you need to check if the new GFP tagged protein is expressed in enough to be visualized. You need to increase protein load per well. You can also add a denaturant, because the tagged proteins may also be folding in a way which will make it more difficult for the antibody to bind. For PVDF membrane, you can dry it up, protein side up, dry up in the air, and this will also fix the proteins to the membrane. If above does not work, you need to try another GFP antibody, unfortunately. And also you need to consider if it was no failure with antibody storage. So to summarize, how to get an intense signal from Western blot using a weak antibody. And here you need to go back to the desk and check. Have I used a proper sample type? Maybe fractionation was required. Maybe I should increase protein load per well. Did I have optimal transfer conditions? Antigen purified antibodies? Did I have appropriate detection reagents? So all in all, it's optimization. So instead of doing blot after blot after blot, you should actually review in details your Western blot protocol. And I would like to share with you the example of optimization. So before it looked like that, afterwards it looked like that. So it's not always an antibody which is to be blamed. In this case, the only parameter which was changed was protein load per well. This antibody was so efficient that it was giving a good signal with 2 micrograms, and the background was diminished. So optimization is a key for obtaining good results here. Advice for beginners: before record a correct band first, before multiplying variations of conditions. This is what I see most commonly, that blot after blot is done, but one should actually review the protocol. And here I'm sharing with you the protocol which we are using on non-characterized antibodies. As you see, we're using high protein load per well. We are blocking only 1 hour room temperature, and we are using highly concentrated primary antibody, 1 to 500, 1 to 1000 overnight, and highly diluted secondary, no longer than 1 hour. This protocol, of course, most likely will generate a lot of background, but the most important thing will be to see a target protein. And once we see it, that it comes versus a good control as mutant, for example, of overexpressor, then we can optimize the background. This protocol will save you a lot of time because you will be able to see if there is any binding at all in with the protocol which you're applying. How do you strip and reuse the membrane to detect multiple proteins? Could you talk about the journal requirements for whole membranes? Yes, of course I can do it. Just a moment. Next advice I have for experienced users. Experienced users' mistake is to use the same protocol for all antibodies. And as you have seen, it is actually not correct because it depends upon protein antibody pair. And now, if you still have such blots, you're most welcome to contact us. And a short quiz before I come and answer your question. Common misconceptions. Think for yourself. Other antibodies I have bought from Agrisera work, therefore this one should work too. True or false? False. The antibody works for species A, so for sure it's going to work for species B. Is it correct assumption? No. The same protocol can be applied for all antibodies. You know it's not true. Antibody gives a good signal in Western blot, so therefore it should also work in other techniques. That's actually not true because it depends how the protein is exposed, and it may be different between immunohistochemistry and Western blot. So remember, one technique, one validation. You cannot assume that antibody which is working in Western blot, it's going to be suitable for other techniques. It has to be experimentally proven. And if you're curious, would like to learn, we are posting new tips on our technical blog. And we are also now coming to the publication issue because there's no worldwide standard, as you know. And this means that more than a half of antibodies used in research cannot be properly characterized, unfortunately. So you should always include antibody name, product number, and supplier. This is extremely important. If possible, also include these data, which, as you see, can be helpful for the end user. Of course, if it is a commercial antibody, this should be already specified on product information sheet. As to your question about requirement of a whole membrane, I actually agree with it because I'm also myself annoyed with seeing only bands of target protein, and on Agrisera product information sheets, you will find whole membrane blots in most of the cases. And the reason for it is that we would like to show you how the antibody is reacting with the given Western blot protocol. In some cases, there is a window for optimization. So, for example, if too much protein was loaded, and that can be optimized later. But for targets of low abundance, there will be other bands, most likely. Therefore, it is extremely important to include reliable controls. And I'm not meaning actin here, I mean a mutant or an overexpressor or a cellular fraction. So I actually agree, and I'm happy that whole membranes are going to be included in publications if this is the case. So we need to help to increase reproducibility, because unfortunately lack of worldwide standard is leading to too little data on which antibodies were used in which experiments. There are free resources which I would like to welcome you to use. We have educational posters which you can receive from our UK distributor, or you can download them from our website. There's also a community resource, the Global Plant Event calendar, which we run together with American Society of Plant Biology for the 5th year. You will find hundreds of conferences, free webinars and workshops which are listed there, and already for the next year, of course. There's also a possibility to use free images of model species and different pathways, which you can find on our website. If you would like to receive, if you're not from UK, of course, and would like to receive our newsletter, you can sign up for it. And here's a surprise. We offer over 100 free antibodies to different targets. Some of them are to epitope tags in return for results. You can review the list from the PDF and let us know in case there are any antibodies of your interest. Antibodies in our catalog are validated by 2 independent laboratories, and that effort is conducted in collaboration with laboratories worldwide, as we do not have access to specific mutants. And you are also always welcome to come with suggestions for antibodies which you would like to see in our catalog, because that catalog, which is celebrating 25 years next year, has been built together with community, with scientists which were coming with suggestions. And if you are from UK, you are contacting 2B Scientific for any questions regarding our products.